This is Unit 8, Molecular Genetics, 8.1, Foundations in Genetics. This unit is going to seem a little shorter, but it's pretty full. Lots of stuff going on. Today we're going to be looking at some people, what they contributed. So as far as my class is concerned, you need to know last names, what they contributed to the field of genetics. So let's talk about these folks. So what is genetics? First of all, genetics, study of hereditary, study of genes. Gene is just like a single unit of DNA that codes for a particular protein. This is how one trait is passed from one generation to the next, or a trait is passed from one generation to the next. We're going to be looking specifically in the next unit concerning Mendelian genetics, like probabilities and that sort of thing. So this unit is more just the how or the why behind genetics, I guess, like the how DNA is replicated and how proteins are made, all that business. So we're going to talk about some people, again, that are pretty important to the field of genetics. We're going to start with Frederick Griffith. Frederick Griffith was a doctor, a military doctor in World War I, and did some other things in World War II. And he is noted for discovering this concept of bacterial transformation. He kind of discovered it on accident. And bacterial transformation is the idea that a bacteria can take up DNA from the outside and be changed by it. Let's look how he discovered it. What do you need to know here? You need to be able to tell me about this experiment and why it's significant. All right, so he basically injected some mice with some bacteria that he knew were relatively harmless, injected them into the mice, and the mice lived. Shocking. And that we'll call that the rough bacteria. For the smooth bacteria, he injected those into the mice. He knew those bacteria to be virulent and to cause pneumonia, so he injected them into the mice, and the mice died. Again, shocking. Why would he do this? To establish control, right? Well, he killed those smooth bacteria. How did he do that? Well, he just boiled them. He killed them and injected those bacteria into the mice, and the mice lived. Right, and so what did he prove there? He proved that those bacteria need to be alive in order to cause damage to the mouse, which makes sense. And so in the next step, he took the rough strain, which didn't cause death. He took the heat-killed smooth strain, which also didn't cause death, combined them, and the mouse died. Why? There was something about the mixture of the two. And so what he said is, he said there was some sort of change agent or transformational agent that caused the rough strained bacteria to be changed into bacteria that would be harmful. And Griffith wasn't able to quite put his finger on it, right? He wasn't able to understand what was the thing that caused them to change. Of course, we know that to be DNA today. And so that's where you get the idea of transformation, which just means to change. So moving on. Oswald Avery is next. We don't have a picture of his experiment. I'm not that concerned that you know his experiments. We just have a picture of him like doing some science-y stuff in the background. And so what was he noted for? Well, he redid Griffith's experiments. He reworked them and seeking to answer particularly the question, what is the transformation agent? And so Avery actually isolated DNA as the change agent that one organism was passing down to the next generation in order to pass this kind of virulent phenotype or virulent trait on to the next generation. And so biology wasn't convinced, though, because, science moves pretty slowly. It's like a giant ocean-going vessel that turns very slowly. So we needed more evidence. Well, we got it. Hershey-Chase comes along. Alfred Hershey, Martha Chase, they did their famous experiment, and the purpose of their experiment was very similar to Avery's, to prove definitively that DNA is the change agent, that it's passed from one generation to the next. Codes for hereditary information and so forth. How they do that? Well, they use these things called bacteriophages. So a bacteriophage, you see there up in the top, these are like viruses, and specifically they attack bacteria. And viruses are very good vectors for this particular experiment because the two molecules in question were, okay, is it proteins that are passed from one generation to the next, or is it DNA that's passed from one generation to the next? And bacteria are just made of those two things, or viruses are made of those two things, protein and DNA. And so they coated some of the viruses with sulfur and the sulfur is going to cling to the proteins because some proteins have sulfur in them, or some amino acids have sulfur in them. And so those viruses, you can see there on the left, the viruses infected the bacteria and they went on about their way. And then one generation to the next, they were not able to find those radioactive sulfur particles because proteins aren't passed from one generation to the next. On the right side, they coated the DNA with radioactive phosphorus because DNA has phosphate in it. And the viruses infected the bacteria, and that radioactive DNA was found in one generation to the next, showing that DNA is the thing that is passed down from one generation to the next. That's Hershey-Chase. Pretty important experiment. It's proved definitively that DNA is the hereditary agent. Very important. I think this was early 50s, maybe, when this happened, just to give you some ideas. So a couple other guys that we need to talk about, or people that we need to talk about. Chargaff, or Erwin Chargaff, came up with this rule, Chargaff's rule, which is basically the idea that the percentage of purines in a cell is going to be percentage similar to the percentage of pyrimidines in the cell, or equal to. And what are purines? Purines are A and G, or adenine and guanine. Pyrimidines are thymine and cytosine. And so specifically, he said that the percentage of adenine, or percentage of A, is going to be equal to the percentage of T. And so what is he showing? He's showing that these two are always found together, right? And C and G are always found together as well. And if you know the percentage of one, you can know the percentage of the other three. How is that possible? Well, let's say if A is 12% of the nucleotides in DNA is A. Well, what's T? It's 12. Add those together, you get 24. How much is left? 76. Divide that in 2, 38. So G and C are both 38. Third grade math. You might see that on a test. Rosalind Franklin is next. So Rosalind Franklin, she was doing work with X-ray crystallography, which was kind of crystallizing these molecules and seeing them in a different way, being able to see really small things with using microscopy, however you pronounce that. And so she was doing this with several different molecules, and particularly was working on DNA, and was trying to discover the structure of DNA, like the three-dimensional structure of the molecule. And so she was applying her techniques to this. This was mid-50s as well, maybe early 50s, late 40s, when she was doing this work. And she took a picture of the DNA, which is a very famous picture now. And so if you can imagine, like looking at the DNA, right? DNA, we know what DNA is, double-stranded. And if you were to kind of look down the tube at the DNA, kind of like you would look down a straw, right? What would you expect to see? Well, this is what she saw. And so from this picture, she was able to deduce that DNA had this spiral kind of shape to it. Unfortunately, she was unable to receive any credit from this. She had some illness at the time, and also there was some shenanigans going on with her her partner and some other people that she shared this information with, and they kind of like pushed her out of the discovery, which is sad, and was not recently, but was discovered that she had a lot more to do with this discovery. But the two that are credited with discovering the shape of DNA are Watson and Crick, and you don't need to know their first names. You'll probably always see them together. And so their claim to fame is that they did discover that DNA is this double helix formation, which means that there's two structures and they're helical with each other, spiraling around one another. They created this giant 3D model of it, which is good. And I think it's important to talk about this whole idea with them and Franklin, because a lot of times you'll hear people say, well, they stole her work or whatever. It's not really how science works. Yeah, there was definitely some indiscretion there, and they could have worked more to give her more of the credit. But science is constantly building upon the work of others, and that's just a norm. Unfortunately, in those days, it was not common for women to be a part of this sort of thing, and so that's why it's interesting that you have Rosalind Franklin here, and of course Martha Chase, here, making these kind of discoveries. So it's a good thing. But yeah, so these two guys actually won the Nobel Prize in 1953 for making that discovery.